everyone, welcome to the Chapter 9 video, which is all about muscles. So first off, why should I care? Muscles control everything you do. If you like to move, breathe, and live, if you like to eat, all of that requires muscles. So that's why you should care. There are 3 types of muscle. Hopefully this is a review from Chapter 4. First type is smooth muscle. It's found in hollow organs. Its main function is moving things through the digestive tract. It is involuntary. Thank goodness we don't have to think about it. This type that's more branched is cardiac muscle. It's found in the heart. It pumps blood. It's also involuntary. And then the third type is skeletal muscle, which is the main focus of this chapter, is skeletal muscle. It is found attached to bones, it causes movement, and it is voluntary. We consciously control this type of muscle. So let's look at some functions of skeletal muscle. The first one is movement. This picture is representing movement, and that's probably its main function. Another thing it does is support soft tissues. If you didn't have muscles holding all your guts in, well, they'd spill out like this, guys. Another thing it does is guard entrances and exits, so it controls eating and also being. Another thing it does is help us maintain posture. This guy is standing with excellent posture. And another function is maintaining body temperature. When we get cold, we shiver, and that generates heat. Alright, our muscles are organs. Remember, an organ is 2 or more tissues working together. And if we look at a muscle, we see that it has connective tissue around the outside. This yellow thing is nervous tissue, and then of course it has muscle tissue. And if we look at a section through that muscle, it would look like this. And we see all around the outside is surrounded by what's called the epimysium. Epimysium. And then the muscle is divided into these things which are called fascicles. They're not cookies, they're fascicles. And if we look at one fascicle, we see that it's surrounded by a perimysium. That's P-E-R-I-mysium. And then a fascicle, this particular fascicle contains 8 individual muscle cells. And they're also called muscle fibers because they are so long. They're some of the longest cells in our body. And then if we look at a close-up of one cell, we'll see that it's surrounded by the endomysium. That's E-N-D-O, mysium. That protects and electrically insulates each cell. And that's important because the muscles that control your middle finger are right by the muscles that control your pointer finger. And you don't want to be accidentally raising your middle finger when you are trying to raise your pointer finger, or you might get beat up. So that's important. These 3 connective tissues that are around muscles, they fuse at the end of a muscle to form a tendon, such as your Achilles tendon. The reason muscle cells are so long, and the reason they have more than one nuclei per cell, is because of the way they're formed. They're formed when several of these myoblast cells fuse together to form a large multinucleate muscle cell. And some of these myoblast cells will remain around in the mature muscle cells, and they'll help with muscle repair if that muscle is ever damaged. Alright, now we're going to look at muscle microanatomy. And Interactive Physiology has a great section on this. If you bought a new book, that disc came with it. If you have the ebook, you can access that website. It's the same login as your Mastering A&P login. And it will take you through all the different organelles of a muscle. So here is a muscle cell. And there are some unique organelles that they have. These rod-shaped things, and if we look even closer, we can see better. So now we're looking at just a section. This rod-shaped thing here is a myofibril. They are the contractile organelles They're made up of myofilaments, and they make up most of the volume of the muscle cell. So you won't see that organelle in any other cells. Then the next one is this blue stuff here, which is called the sarcoplasmic reticulum. If we look even closer, there's the sarcoplasmic reticulum, and you can see it surrounds the myofibrils. And it is the smooth endoplasmic reticulum of muscle cells. They surround myofibrils, and if we look closer, they have special calcium channels. They store and release calcium. And we'll see later on why calcium is necessary for muscle contraction. Okay, then another unique organelle is the sarcolemma, which is the plasma membrane of the muscle cell. And you'll see it has these holes in it. We'll talk about them in a second. These holes lead to these yellow things, which are called transverse tubules. They're a continuation of the sarcolemma, or the muscle cell membrane, and they bring electrical signals deep into the muscle cell. So one of the jobs of the plasma membrane is to generate an electrical signal, and we'll talk about that process in a bit. But that electrical signal then spreads down the T-tubules, and that way the myofibrils deep in the muscle cell get the signal to contract at the same time as the myofibrils at the surface. So we're going to look closer at myofibrils now. They are made up of sarcomeres. Sarcomeres are the smallest functional unit of a muscle, and they're made up of what we call thick and thin filaments. The thick filaments they're blue in this figure. The thick filaments are myosin, M-Y-O-S-I-N, and the thin filaments are made up of actin. And if we look here, this is a Z line that is the connection between adjacent sarcomeres. So a myofibril is a series of sarcomeres all connected together, and they're connected at the Z line. The M line will be right here in the middle of each sarcomere, connects the central portion of each thick filament. The A band is right in here. The A band contains the thick filaments, so it looks dark under a microscope. The I band is right in here. It contains only the thin filaments, only the actin, and no thick filaments. And then the H band is this region right here where you have thick filaments but no thin filaments. And then it's not labeled on this picture, but there's a zone of overlap here. That zone of overlap is where you have both the thick and thin filaments overlapping. Alright, so again, here's another look at our sarcomere, the thick filaments, the myosin, and then in red you have the thin filaments, or the actin. So, and again, this shows all the parts of a sarcomere. You've got the Z lines, the I band, the H band, the A band, and on the top there is what a real skeletal muscle looks like. This is a very high magnification. Alright, so the thin filaments, composed primarily of actin. If we look even closer at them, we'll see all the components. Each actin molecule has an actin site, which in this figure is shown in yellow, and that combined to myosin. This protein in the middle that's holding everything together is called nebulin. Nebulin connects the actin molecules together. You see on here G-actin, which each individual ball would be globular actin, but together it makes up F-actin, which is filamentous actin, which does something for us. Also in the thin filament, this braided protein here is called tropomyosin. It covers the binding sites and it blocks myosin from binding to actin. And we'll talk about how that works in a minute. Then these other molecules here, troponin. You can see there's troponin molecules. attached to the tropomyosin. It binds to tropomyosin and actin, and it has a calcium binding site. And we'll see how that works in a bit. So that's the thin filaments, primarily actin, but also you have nebulin, you have tropomyosin, and troponin. Then if we look closer at a thick filament, we see that they have a core of titin. It's a very strong protein, and it prevents the muscle from being overstretched. The thick filaments are composed mainly of myosin molecules. Here's one myosin molecule down here. They have a tail, and then they have two globular heads. They look sort of like a golf club. The myosin head, this is what binds to actin, and it rotates or pivots to cause contraction. And if you want to learn more about those individual proteins, again, go to that interactive physiology disc. So next thing we're going to talk about is the sliding filament theory. And when a muscle contracts, here we have a relaxed muscle and here we have a contracted muscle. When a muscle contracts, the thin filaments slide past the thick filaments. And the H-band, you can see the H-band and the I-band gets smaller, and the zone of overlap gets larger. And the Z-lines come closer together. You can see here on the right, the Z-lines are much closer than here on the left. And so with all of these individual sarcomeres shortening, that leads to the whole myofibril shortening, and all the myofibril shortening lead to the muscle shortening, and that's how we get contraction. Alright, some review questions. Which organelle is the contractile unit of a skeletal muscle cell? And that would be C, the myofibril. Then what organelle functions to store and release calcium in a skeletal muscle cell? And that would be B, the sarcoplasmic reticulum. The plasma membrane of a skeletal muscle cell is also called the, and that would be A, the sarcolemma. The organelle that sends an electrical signal deep into a skeletal muscle cell would be the transverse tubules. This protein has a calcium binding site. The only one of these that has a calcium binding site is troponin. And this protein blocks myosin binding sites on actin, and that is the job of tropomyosin. This protein has a head that pivots to cause contraction, and that is the job of myosin. Alright, skeletal muscle fibers and neurons have excitable plasma membranes that produce and carry electrical impulses called action potentials. And because the plasma membrane is selectively permeable, ions cannot cross it. There is a charge separation across the plasma membrane which represents potential energy. And we can see that in this figure. The potential energy difference across the plasma membrane is called the membrane potential. And for a resting cell, the inside is usually negative compared to the outside, about negative 70 millivolts. And so this figure shows that the inside of the cell is negatively charged. And this is due to ions normally cannot cross the plasma membrane, but we do have some leak channels that will allow potassium and sodium to cross the plasma membrane. And I want you to remember this figure. Your muscle cells, well, all your cells really, are like bananas in salt water. And we know bananas have lots of potassium, and the Salt water has lots of sodium chloride. Sodium and potassium are the main ions we're going to be concerned about with the generation of action potentials. And inside the cell you have a high potassium concentration, outside the cell you have a high sodium concentration. So that's why here the amount of sodium outside the cell is higher than the amount of potassium inside the cell. That's why the inside of the cell is negative compared to the outside. And also you have lots of negatively charged proteins inside the cell. So the charge difference is due to different concentrations of ions. Sodium is higher outside the cell, and potassium is higher inside the cell. And we also have lots of negatively charged proteins inside the cell. Now I mentioned back here there are some leak channels that are always open, and at rest there are more potassium leak channels open than sodium leak channels. And we're going to talk about this more later. We just need to briefly introduce it right now. But the way things get through a plasma membrane, the way ions get through a plasma membrane is through ion channels. This is a chemically gated ion channel that will be closed when nothing's bound to it, and when a neurotransmitter is bound to it, it will open and allow ions to pass through. So that's one type of ion channel, chemically gated. And we have some of those in muscle, but we also have what are called voltage-gated ion channels. And neurons and muscle cells have these voltage-gated ion channels in their plasma membranes. And these channels are open or closed by changes in the membrane potential. And the way we think these work, remember they're proteins, and proteins are made up of amino acids. And there are 20 different amino acids. Some of them have positive charges, some of them have negative charges, some are acidic, some are basic. They all have different structures. But in this region of the protein, you have lots of amino acids with a positive charge. So this part of the protein is positively charged. And normally the inside of the cell is negative, so remember opposite charges attract. So these stay down and this channel stays closed, the ions can't go through it. When something happens to allow some positive charges to come into the cell, usually one of those chemically gated channels that we just saw opens and allows positive ions to come in, those positive charges will repel the positive charges here and open this ion channel, and now ions can flow through it. So that's how a voltage-gated ion channel works. Now we're going to talk a little bit about action potential generation. We're just going to introduce this right now. So at rest, the cell is about -70. A charge reversal begins with a small increase in sodium permeability up to a set threshold. And this is the threshold right here. It's different for different cells, but about -55. At threshold, those voltage-gated sodium channels are going to open, and positively charged sodium ions rush into the cell. So that's the point where enough positive charges, the threshold is the point where enough positive charges come in to open this channel. And then that allows a lot of sodium ions to come into the cell. Sodium ions rush in because, remember, the sodium concentration is higher outside the cell. The inside of the cell then becomes positive, and it's said to be depolarized. So now we get up here to about +30. Depolarization peaks at about +30 millivolts, at which point voltage-gated sodium channels close and voltage-gated potassium ions open. And now, since potassium is higher inside the cell, positively charged potassium ions are going to leave the cell. And as those positive charges leave, the inside of the cell becomes more negative. the membrane potential drops below the resting potential as voltage-gated potassium channels close. And during the refractory period, another action potential cannot be generated because ion conditions are being restored to their resting states. And we'll come back to action potentials in Chapter 11 when we talk about neurons, and we'll talk about them in more detail there. And then an action potential.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neural transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in 2 ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
Okay, so if we want to move a muscle, the first thing we have to do is think about it. And we think about it, and from our brain, it sends a signal down, what's called a motor neuron, and that's what causes the muscle to contract. So a skeletal muscle fiber contracts when stimulated by a motor neuron. And each skeletal muscle cell, here's one cell, has what's called a neuromuscular junction, where the end of the motor neuron, or we also call that the synaptic terminal, because that's where it ends. The synaptic terminal comes close to the motor end plate of the muscle cell. And the motor end plate is right here where the synaptic terminal contacts the muscle cell. And if we look closer at this, we can see they're separated by what's called the synaptic cleft. They're not actually touching, but they're very close. The synaptic terminal contains the neurotransmitter acetylcholine, or we abbreviate that as ACH. This is a chemical signaling molecule. And when an action potential, here's our action potential arriving, it's red in this picture. That's an electrical signal. An action potential is an electrical signal. And when that reaches the synaptic terminal, acetylcholine is released, which we see in this picture. Acetylcholine is being released. Acetylcholine diffuses across the synaptic cleft and binds to receptors. So here's the receptors on the muscle cell, on the motor end plate specifically. And these acetylcholine receptors are ion channels, and they allow sodium ions to rush into the cell. Remember, the sodium concentration is higher outside the cell, so when sodium channels open, sodium wants to go into the cell just due to diffusion. And this is going to generate an action potential, and that action potential is going to spread along the sarcolemma. Here it is, spreading along the sarcolemma and down the T-tubules. And when here it is going down the T-tubules, and when it gets to the sarcoplasmic reticulum, it's going to cause calcium to be released. And then we have a very important enzyme, which we can see back in this figure. This is acetylcholinesterase right here, these banana-shaped things. And it breaks down acetylcholine to prevent continuous contraction. Okay, so if you prefer these steps in a list, here it is. The action potential reaches the end of the nerve. Synaptic vesicles fuse with the plasma membrane and release acetylcholine. Acetylcholine binds to receptors on the muscle, opening ion channels. The inward flow of sodium generates action potential in the muscle. And the action potential travels down the sarcolemma and down T-tubules. And then calcium is released from the sarcoplasmic reticulum. Alright, so that's how we get the action potential, that's how we get the signal from our nerve to our muscle. So now we're going to watch a movie about excitation, contraction, coupling. Going back to, let's go to this page. And back to AMP Flicks. And so you can watch these movies again on your own by going to this Mastering AMP website.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with the calcium-storing membranous network known as a sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, Calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
Okay, so that's a close-up of the last part of that process we just talked about, how calcium is released. And then we're going to see in this next section what calcium does. But first, some review questions. So acetylcholine is released from the synaptic terminal in response to a or an, and the correct answer is C, action potential. The effects of acetylcholine are terminated by, and the correct answer is A, acetylcholinesterase. The end result of excitation contraction coupling is, and the correct answer is calcium release, and I can pretty much guarantee that question is on your test. So now we're going to look at what happens after calcium is released, and that's the contraction cycle. So we're going back to AMP-Flix, and here it's called the cross-bridge cycle, contraction cycle, same thing.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross-bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released, and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin binding site on actin.
Okay, so that's the contraction cycle. And if we look at a muscle up close, you can see the thick filaments and the thin filaments, and these things reaching out. Those are called myosin cross bridges. But it's hard to tell what's what in that picture, so we're going to go to a diagram. So the contraction cycle. In a resting sarcomere, such as this one, tropomyosin is blocking the myosin binding sites on actin. So myosin wants to bind to actin, but tropomyosin is blocking. But when calcium enters, it binds to troponin. Troponin changes shape. Here's troponin. Troponin changes shape, and it moves tropomyosin, exposing the binding sites. So the first step is active site exposure, and this, depending on which book you read, this is part of excitation, contraction, coupling, or part of the contraction cycle. And then once the active sites are exposed, then we have cross-bridge formation. Now you can see myosin is binding to actin. Then the next step is myosin head pivoting. The myosin head rotates or pivots towards the M line, pulling actin towards the middle. And as it pulls actin towards the middle of the sarcomere shortens. And the next step is cross-bridge detachment, and this requires ATP. So when ATP comes in and binds to the myosin head, it changes shape and it's going to detach from actin. And then we have myosin reactivation, where the ATP is split and the energy that was stored in that bond is used to reactivate the myosin head, and then it's ready to go again. And the process will keep going as long as calcium is present and as long as there's enough ATP. That process will keep going. So if you prefer a list, first step is active site exposure, where calcium binds to troponin, changing its conformation. Troponin moves and pulls tropomyosin away from the myosin binding sites on actin. Then we have cross-bridge formation. Then the myosin head pivots. That's also known as the power stroke. Then we have cross-bridge detachment. This is the step that requires ATP. And then finally myosin reactivation. So that is the details of how a muscle contracts. I would encourage you to use the interactive physiology disc if you want more review. But now we're going to move on to talk about muscle tension. So this is an overview of the steps. So the first step is neural control. We have to have the action potential coming from the neuron. Then excitation, contraction, coupling happens where calcium is released. And then we have the contraction cycle, the myosin head's pivoting, and that's what leads to the contraction of the muscle. And that's what produces tension. So looking at this figure, it shows that the muscle tension is greatest when a muscle is stimulated at its optimal length. This right here is the optimal resting length. And you can see over as it goes away from this optimal length, the tension produced decreases. If you stretch a muscle too far, this is actin here and this is myosin. If the actin and myosin can't overlap, the myosin can't bind to actin and you can't generate any tension. So that's why it drops as you get further away. As it comes closer together, there's more overlap, tension goes up. At this point, there is maximum overlap of actin and myosin. This is where the maximum number of cross bridges can be formed, but it's before the thin and thick filaments start to overlap. So as they start to overlap, that's when tension starts to go down, and they can only get so short before they crash into each other. So that's why tension goes down when it gets too short. Okay, this is what is called a muscle twitch, and it's the response of a muscle to a single stimulus. And smaller muscles, you can see this is the eye muscle, a very small muscle, and here's a big muscle in your calf. You can see that smaller muscles contract and relax faster than larger muscles. And if we look at the phases of a muscle twitch, there's a couple different phases, 3. First, you have what's called the latent period. So this is where the stimulus happens. And the latent period is when excitation contraction coupling happens. You can see this is only 2 or 3 milliseconds. But during this phase, no force is generated. No force is generated. Then we have the contraction phase. This is when we have the cross-bridge cycling. So actin and myosin are interacting during this phase. And this is when we generate our peak force. And then during the relaxation phase, calcium levels drop. And because of this, tropomyosin starts to cover the myosin binding sites on actin, so cross-bridge cycling stops, and the tension goes to 0. Alright, and the peak tension produced by a muscle depends on the amount of tension produced by each muscle fiber and the number of muscle fibers stimulated. And this figure right here is showing us what's called TREP. If a muscle cell is stimulated right after it has relaxed, the next contraction will produce greater tension. So here's the first stimulus, and then if next stimulus is given right away, it's going to increase the tension of the next stimulus, and if it happens again, it increases further, and it increases until we get up to our maximum tension. We believe this is because there is still some calcium out in the myofibrils. Not all of the calcium has been pumped back into the sarcoplasmic reticulum. So when more calcium is released, now the calcium concentration in the muscle is higher, and it continues to build up until we get to maximum tension. This figure shows us what's called wave summation. So if a second stimulus arrives before the relaxation phase ends, the next contraction is greater or stronger. And it's due to the same reason that the calcium has not all been pumped back into the sarcoplasmic reticulum at this point. So this next action potential is going to release even more calcium, and as the calcium concentration builds up, the tension gets stronger. This figure shows what's called incomplete tetanus, when a muscle produces almost peak tension due to rapid stimulation. So its maximum tension is up here, and it's doing wave summation, and it keeps stimulating this muscle very rapidly. This would be called incomplete tetanus because we haven't quite reached the maximum force. This figure shows complete tetanus, when the muscle is stimulated so frequently that it cannot relax. Maximum tension is generated, and that's you've got the maximum amount of cross-bridge cycling happen, the maximum overlap of actin and myosin. So that's the maximum force this muscle can generate. So a review question. Muscles produce the greatest force at an optimal length because, and the correct answer is because there is maximum overlap of actin and myosin. When a muscle is stimulated a second time before it fully relaxes and increased force is produced, this is known as, and this would be wave summation. And smaller muscles contract faster than large muscles. Okay, a motor unit is all the muscle fibers controlled by a single motor neuron. And small muscles that need precise control, such as the muscles in your eye or your hand, they have small motor units. And large muscles that generate lots of force, such as your leg and back, have large motor units. And here is an example of some motor units. So here we see 3 different motor neurons, and they're color-coded in this example. So we have a red motor unit, which is this motor neuron, and all these red muscle cells, and then the blue motor neuron contacts all the blue muscle cells, and the purple motor neuron contacts all the purple muscle cells. And during a sustained muscle contraction, these motor units will rotate being active. So first the red one will be active, and then as it starts to relax, the purple one will contract, and then the blue one will contract, and they will rotate. So that allows the muscle to rest, and that way it can contract for a long period of time. Alright, we have something called muscle tone. Even in a relaxed muscle, some motor units are contracting. This is subconsciously controlled, and it keeps our muscles healthy and ready to contract. This also stabilizes joints and maintains posture. Now, this next figure is going to teach us about muscle tension. So muscle tension can be changed by frequency modulation or recruitment. And this figure shows recruitment. Recruitment is increasing the tension by increasing the number of motor units stimulated. So the top figure shows us the stimulation they're using. The bottom figure shows us the amount of tension produced, and this middle figure shows us the number of motor units that are excited. So in this case, if we don't reach threshold, we don't get an action potential, so no motor units are stimulated. At threshold, it looks like 2 motor units are stimulated, so you get that much tension. As the stimulus increases, now we've got 5 motor units stimulated and the tension increases. Now we've got 10 motor units stimulated and the tension increases, and now we have more motor units and the tension increases until we get to what's called the maximal stimulus. At that point, that's the maximum force this muscle can generate. So increasing the stimulus beyond that isn't going to make any difference. And this again shows how motor units rotate being active during a contraction. So this is showing the tension in the tendon, which is produced by the muscle. And the tension stays up here, but you can see the red motor unit is only active here and here. And then during this time it's due to the blue motor unit, and then during these times it's due to the purple motor unit. So we said this already. Muscle tension can be changed by frequency modulation or recruitment. Frequency modulation is increasing the tension by increasing the frequency of stimulation. And then recruitment is increasing the tension by increasing the number of motor units stimulated. There are some different kinds of contraction, isometric and isotonic. So this here is an isometric contraction. When force is generated but the length of the muscle doesn't change, this is an isometric contraction. So you can see it's generating force, but it's not strong enough to lift this weight. So it's producing tension, but it's not changing its length. That's an isometric contraction. Then when force is generated and the length of the muscle changes, that's an isotonic contraction. So we produce tension and the muscle shortens. That's an isotonic. And this right here is a concentric isotonic contraction. This is the most common type. The muscle shortens. And then in an eccentric isotonic, the muscle would lengthen. This figure shows that the speed of contraction is inversely proportional to the load. So this shows the distance shortened and also the time. This shows the velocity of shortening with an increasing load, which makes sense. As the load gets heavier, it takes the muscle longer to generate enough force to lift it. So velocity is going to be fastest with a lighter load, slower with a heavier load. Alright, a little review. Changing the tension produced by a muscle by changing the frequency of stimulation is known as frequency modulation. So if you tell a muscle to contract more frequently, it's going to generate more force. A muscle contraction where the length of the muscle does not change, that is called isometric. And a muscle contraction where the muscle shortens is called, and the correct answer is concentric isotonic. Alright, contraction requires energy. And we know our cells use ATP for energy. And it's produced from a couple of different sources. And again, the interactive physiology disc has a great section on this. This figure shows an overview of how ATP is generated. The three sources of energy in a muscle are ATP, creatine phosphate, and glycogen. So ATP, the stores are depleted in about 2 seconds. So we don't store very much of that. You can see that's enough for about 10 twitches. With creatine phosphate, that lasts a little longer. That lasts about 15 seconds, good for maybe 70 twitches. And then glycogen is broken down by glycolysis to produce ATP, and that can produce ATP for about 2 minutes. Then there's another process that can occur. So we're going to use up our ATP stores first, then we're going to use our creatine phosphate, then we're going to use up our glycogen. And at rest, the muscle is using mainly fatty acids. And it turns those fatty acids into ATP through the process of aerobic respiration. Aerobic respiration can produce ATP for hours. It's the most efficient way of producing energy, but it requires oxygen. And a muscle cell at rest can build up reserves of ATP. At moderate activity levels, a muscle cell gets energy from aerobic respiration. And then at peak energy levels, peak activity levels, the muscle cell gets energy from aerobic respiration and from lactic acid fermentation. So it will break down glycogen into glucose, into pyruvate, and that produces lactic acid as a byproduct. Alright, so which source of energy will last the longest? And the correct answer is aerobic respiration. Next topic is fatigue and recovery. So muscle fatigue is the inability to contract due to changes in ATP levels and ion balance. When there is insufficient oxygen, the muscle converts pyruvate to lactate, which produces lactic acid. Lactic acid builds up in cells and lowers the pH. When oxygen is available, the muscle recovers and converts the lactate back to pyruvate and regenerates our glycogen reserves. And most of this conversion happens in the liver. It's converting lactate. So the lactate leaves the muscle, goes to the blood, and it gets converted back to pyruvate in the liver. During this recovery period, your body is using more oxygen than normal, and this is called the oxygen debt, or the excess post-exercise oxygen consumption. So this is why you have to, after you run a race, you're going to have to breathe hard for a few minutes. You're going to keep taking deep breaths because you're trying to build back up your glucose stores. All right. Okay, next topic is muscle fiber types. And Interactive Physiology, the disc or the website, has great information on this as well. Here's a picture of some different types. This would be the fast glycolytic, the slow oxidative, and fast oxidative, or this is called an intermediate fiber sometimes. Here's what some fast fibers look like. They're relatively large. This eye muscle is all fast fibers. That figure we saw before was a mix. And then you can see the slow fibers. They have a much smaller diameter. This figure tells you everything about them. So the fast fibers, they have a very large diameter. They have high glycogen content because they use lots of glycogen. They have a very low myoglobin content because they don't use myoglobin. They have very few mitochondria. They have few capillaries. They contract very fast, but they also fatigue fast. So these are called the fast fatigue muscles sometimes, or these are fast twitch muscles. Then the intermediate fibers, they have a medium diameter, medium amounts of glycogen, medium amount of mitochondria, medium capillaries. They contract at a medium speed, and they are moderately resistant to fatigue. So everything about them is medium. Then the slow fibers, they have a much smaller diameter, low glycogen. They have high myoglobin. Myoglobin is an oxygen binding protein. And these slow fibers use mostly aerobic respiration to get their energy. These faster fibers, they use mostly glycogen. So, and the intermediate fibers use kind of a mix. So the slow fibers are using almost all oxygen to get their energy. So they have high myoglobin, they have many mitochondria, they have many capillaries to bring them oxygen. They contract relatively slow. I mean, it's fast, but it's slow compared to the fast or the intermediate fibers. And these are fatigue resistant, so they don't fatigue. They can contract all day. So what type of muscle fibers would you expect a world-class sprinter to have a lot of? And a sprinter just has to sprint for about 10 seconds. So they don't need to go for a long time, so you would expect them to have lots of fast fibers. And indeed they do. What type of muscle fibers would you expect a world-class marathon runner to have a lot of? And the marathon runner has to go all day for, well, not all day, but several hours. And they would have lots of slow fibers because those don't fatigue. Alright, last topic is some disorders or changes. So with extensive exercise, muscle cells will develop more myofibrils, larger glycogen reserves, and more mitochondria. and this causes hypertrophy, which is an enlargement of the muscle. So this guy here and me with my little wimpy arms, we have the same amount of muscle cells. His muscle cells are just a lot bigger because he's developed more myofibrils and also more glycogen reserves, more mitochondria. There's probably more blood supply to his muscles as well. When muscles are not used, they lose mass. You may have noticed this if you've ever been in a cast. This guy had a cast on this leg, and you can see the muscle kind of shriveled up. That's called atrophy. Muscular dystrophy leads to progressive muscle weakness. And then we're going to talk about some things that affect the neurons. First, polio. Polio virus attacks motor neurons in the spinal cord and brain. And if the muscle can't get the signal to contract from the motor neuron, then it can't be used. So that leads to paralysis and atrophy. Another thing that affects the neurons is tetanus. It causes sustained, powerful contractions throughout the body, also known as lockjaw. So if your muscles can't stop contracting, like the muscles that control your breathing, you need them to stop and relax so you can breathe in and out. But that's a pretty fatal disease. Botulism prevents release of ACH at the neuromuscular junction. And if you can't release acetylcholine, the muscle can't contract. Myasthenia gravis causes muscular weakness due to loss of acetylcholine receptors or ACH receptors at the neuromuscular junction. And then one last disorder is rigor mortis. This is muscle stiffness due to the lack of ATP. And when you die, you run out of ATP, and so the cross bridges can't detach. They're stuck in the contraction cycle. They're stuck like this. Maybe one up. So the cross bridge is formed, and actually it would do this, but then they're stuck because they need an ATP to come in and bind here so that they can detach. And when you die, that doesn't happen. That's why dead people get stiff. And that is chapter 9.